Okay, this is where we got to last time. So we're going to talk about the two types of immunity, cell-mediated and antibody-mediated. So first type is cell-mediated, and we're now talking about primarily the T cells, cytotoxic T cells. All right, and I showed you this right here. So what you see in beige right here is the way T cells respond. We're going to go through this. But say here's a new antigen that we're exposed to. So all of our lines of defense can respond, and often do, to this antigen. These are general defenses over here. They do not confer immunity. Specific defenses, adaptive, do confer immunity. So what you see in beige here is how the cell-mediated immunity works. All right, so when we talk about cell-mediated immunity, we've got to recognize the antigens. We have specific cells that help do this, help expose foreign antigens to T cells. Our various T cells have specific markers on them. There are a number of different markers on the membranes of T cells, but the two main ones we're talking about, I'll show you. Then we've got something called co-stimulation. This means that when our immune system confronts a new antigen and is prepared to attack and destroy body cells that contain the foreign antigen, there is a built-in safety mechanism to make sure that we don't destroy normal cells. So this is a safety check mechanism. Before those T cells are fully activated to work, there's an internal mechanism that verifies the identity of a foreign antigen and gives the okay for the T cells to go ahead. Now, as far as recognition of antigens is concerned, we have some special proteins that do this in our cells. They're known as MHC proteins. That stands for Major Histocompatibility Complex. There are two classes of them. Class 1 MHC proteins present antigens on the surface of tissue cells, fixed tissue. Class 2 MHC proteins present antigens to T cells in body fluids. So T cells can be exposed to foreign antigens when they come in contact with a fixed tissue cell that contains a foreign antigen, or when they come in contact with a specialized cell that's showing them a foreign antigen or a piece of a foreign antigen that they are specific for. So this is the way this works. In tissue cells, here, say that we have a foreign antigen inside the cell. Here's the cell membrane. Here is extracellular fluid out here. The cell takes this antigen and breaks it down, and in its endoplasmic reticulum, and delivers usually a fragment of that foreign antigen out to the cell membrane. So it's exposed to T cells that are passing by. Now this is important because T cells cannot see inside cells. T cells could not see this in here. They can see it when it's out on the surface of the cell, in the cell membrane. That's why this is important. This presentation of antigen to T cells. The other, and so this is class 1 MHC proteins. The other class of MHC proteins is a specialized type of cell moving in extracellular fluid that picks up foreign antigen, brings it into its cytoplasm, similar to class 1, breaks it down, and presents an antigenic fragment on the surface of its own cell membrane. So that when this cell comes in contact with one of the T cells of the community of T cells that are specific for this antigen, then the process of response to the foreign antigen can begin. So that's what these MHC proteins are for the presentation of antigens. Really, it's fragments of the antigen, not the whole antigen usually. All right, so we have a class of cells that are good at presenting antigens to T cells. They're called antigen-presenting cells, or APCs. APC stands for antigen-presenting cell. So they present antigens to T cells and B cells, which we will come to later. But they do not themselves act against the antigens. The cytotoxic T cells do. So they present them. Now, which ones are the antigen-presenting cells? Well, dendritic cells do, macrophages do, and actually B cells are antigen presenters too. So when we get to antibody-mediated immunity, we'll come back to this and show you how B cells present antigens to helper T cells. All right, so here is a photomicrograph, electron photomicrograph of a dendritic cell. This is one of the class of antigen-presenting cells. So these cells will latch on to foreign antigens and carry them to T cells. They're going to carry them back to, typically they're going to carry them back to the lymph nodes where they can be exposed to T cells. Here's a macrophage that can also act as an antigen-presenting cell. As I mentioned, B cells are also a type of antigen-presenting cells because here is a B cell presenting its antigenic fragment to a helper T cell. Now, the action of this helper T cell is going to be required in order to fully activate this lymphocyte and the clones of it, the ones that are just like it, against this specific antigen. But we'll come back to that later. Now, our T cells have certain markers in their membranes. These are called CD markers. CD stands for cluster of differentiation. There are a number of different markers, but the two that we want to talk about are CD8 and CD4. CD8 markers are present on cytotoxic T cells. Cytotoxic T cells are the ones that destroy tissue cells containing foreign antigen, CD8. CD8 markers are also on the memory cells of this kind of cell. CD4 markers are present on helper T cells, but this type of T cell, helper T cell. So there's a difference between cytotoxic T cell and helper T cell. CD4 is also on the memory cells of helpers. CD4 marker is also on a type of T cell known as a regulator T cell, which I'll explain later. So here now is a display of T cells. So they were formed in the bone marrow. became immunocompetent in the thymus. And now this side over here is dealing with T cells that have CD8 markers. The ones that have CD8 markers are the cytotoxic T cells. These are the ones that do the killing of cells that are affected by foreign antigens. This side over here is also T cells, but these are helper T cells. They have CD4 markers on them. So these are the helper T cells. Now, in order for this cell to be fully activated down here, commonly requires the assistance of a helper T cell. That's why they call them helper T cells. Now, in this case, on this particular diagram here, we have an antigen being presented to a cytotoxic T cell. When this cytotoxic T cell came out of the thymus and had not yet met any foreign antigen that it was specific for, it is said to be naive. So this is a naive cytotoxic T cell. Now it meets the antigen it's specific for. It's going to now become activated. So an antigen presenting cell is presenting usually an antigenic fragment to this cell and its own kind. Every other T cell that's like this one, that's specific for this antigen, can respond to that antigen. It's not just this one. And so now we've got activation and we've got cloning, making more of its own kind. Now we have fully operational cytotoxic T cells down here. These are the ones that are going to do the killing. Now, right up here, with activation, we also have the formation of some memory cells. These are just like these. But these are going to be held in reserve. All right? So that after these cells take care of this, the cells that contain this foreign antigen, and say the crisis is over, the response is over, if we meet that same antigen again down the line, we already have some memory cells that have identified this antigen. They've built up their numbers, their population. So whereas the initial reaction is going to have some delay in it when these lymphocytes meet this antigen for the first time, if and when they happen to meet them again, the reaction is going to be swift. It's going to be immediate and much stronger than the first time. While this is happening over here, this is happening over here. We're making helper cells that assist in the activation of cytotoxic T cells, especially if they're met in extracellular fluid. Now, I'll show you this too later, but this side is helper T cells that have this marker on them, their cell membrane. But the regulator T cells, the ones that put the brakes on the whole immune reaction, called regulator or Treg, they have the same markers, CD4. Now, this thing about co-stimulation, this is that built-in safety check mechanism that I told you about. It is a safety mechanism that ensures that T cells do not attack normal cells. All right, so this happens via some stimulatory proteins that are called cytokines. When we're talking about nucleated cells in fixed tissues, the T cell first must bind to a second site after it attaches to the primary site that contains the antigen fragment. Before it can attack the cell, it must bind to a second site on the cell membrane. That's essentially telling the T cell, it's okay, this is one you need to take out. Now, on our diagrams in our text, this is not shown, but it has to bind to a second site on an affected cell before it can be fully activated. In tissue fluids, the T cell is first assisted by an antigen-presenting cell, which is presenting the antigen to the T cell, and helper T cell has to come along and finish out the co-stimulation process. This is the safety check right here, the assistance of a helper T cell before they will attack cells that they need in tissue fluid. So again, this is what happens in nucleated cells. I showed you this before. This is where the T cell will bind to this, but it must bind to a second site on the cell membrane, which, as I say, is not shown on this particular diagram. But that's what the co-stimulation process is. Now, once a foreign antigen presence is confirmed, the T cells that are specific against that antigen will attack any cells that are carrying that antigen wherever they are found by those T cells in the body. If co-stimulation does not occur for some reason, the T cell will leave that cell alone. It'll be tolerant to it. Another thing about these co-stimulation proteins is that they promote cell division and differentiation in those T cells. This is how T cells, cytotoxic T cells, work, and how they destroy cells that contain foreign antigens. So, this would be the foreign antigen here. This would be the cell membrane of the T cell right here. So, the T cell is releasing molecules called perforins that create holes in the cell membrane. Enzymes from the T cell then enter the cell and break it apart internally, and it dies. This is direct cell-to-cell contact with cytotoxic T cells. Here is an electron photomicrograph of a cytotoxic T cell attacking a cancer cell. So it's doing, up here, although you can't see it, it's doing what that previous slide showed you. All right. So, again, for T cells, we have our cytotoxic T cells, we have our helper T cells. In this case, you have an antigen-presenting cell presenting an antigenic fragment to a cytotoxic T cell. You have an antigen-presenting cell presenting an antigenic fragment to a helper T cell and leading to their activation. Now, both kinds have memory cells. So we have memory cytotoxic Ts, we have memory helper T cells. Now, the regulator T cells are a class of T cells. So you have cytotoxic T cells, you have helper T cells, and you have regulatory T cells. The regulatory T cells are not as numerous, and they come into play after the cytotoxic T cell action is initiated. So what they are doing is they are depressing the T cell response, the cytotoxic T cell response. They also suppress B cell response, but they're essentially putting the brakes on the immune response. They're slowing it down. They're fewer in number than cytotoxic T cells. There's a lag time, as I mentioned. They're not formed right away. They're formed a little bit after the cytotoxic T cells. The presence of these is thought to be an important factor in preventing autoimmune reaction because they suppress any self-reactive lymphocytes that might be present in the body. So here is our regulatory T cells right here. They are sort of an offshoot of memory cell, a helper T cell right here. So here's where our regulatory T cells are, right down here. Speaking of helper T cells, helper T cells are extremely important and absolutely essential to normal function of both T cells and B cells. They help both sides. Their major function is stimulation of proliferation of other T cells and of B cells that have already become bound to foreign antigen. Notice that without helper T cells, there can be no effective immune response because of their involvement in the activation of all other immune cells. So that's why they're called helper T cells. Again, here's your helper T cell line right here.
And that's what HIV attacks?
Exactly. HIV attacks helper T cells. That's exactly right. So what all do these helper T cells do? Well, as far as the cytotoxic T cells are concerned, they provide co-stimulation to them. They help activate the cytotoxic T cells. They promote proliferation of cytotoxic T cells. And they essentially do the same thing for B cells. Both sides. Also, as far as our general or non-specific defenses are concerned, they activate macrophages, they activate natural killer cells, they stimulate the inflammatory response. So these little cells, these helper T cells, very important. Here is the way helper T cells work in cytotoxic T cells, the CD8 kind. Here happens to be an antigen-presenting cell. Oh, here, antigen-presenting, no, here we are. Antigen-presenting, the beige is T cell. All right, here is our cytotoxic T cell. Here is an antigen-presenting cell presenting an antigenic fragment to the T cell that's specific for this antigen. But alongside comes a helper T cell here. And this helper T cell is going to stimulate this APC to co-stimulate this cytotoxic T cell right here. That is not shown on this diagram right here. The helper T cell is also secreting some stimulatory proteins, interleukins, that's what this stands for, that facilitate this whole process of activation of cytotoxic T cells. So you see here, in this case, in the type 2 MHC presentation, T cells are absolutely important here. This T cell cannot be activated just by contact with this antigen-presenting cell. It has to have the assistance of a helper T cell right here. Now, in the B cells that we'll get to later, you'll see that we have to have a helper T cell helping out here, because this B cell is presenting a foreign antigen. Remember, B cells are antigen presenting cells. It's presenting a foreign antigen to a helper T cell. And that helper T cell is secreting some stimulatory proteins, interleukins, that are going to lead to the full activation of this B cell. And this B cell is then going to produce plasma cells, which produce the antibodies. We'll come back to that later. All right. Now, just a word about homograft rejection and immunosuppression. A homograft is a transfer of tissue from one member of a species to another member of the same species. So organ transplants are homografts, all right, or allografts, but homografts. So a kidney transplant is a homograft. It's a transplant of a kidney from one human to another. Now, in this process, there is a screening procedure where you try to match tissues as closely as possible. But in spite of all the preliminary tests and the closest matching possible, can you ever get a perfect match?
No.
Only in one kind of case.
Twins.
Identical twins. All right, so you can get close, but you can't get perfect. So that's the reason that organ recipients have to be on immunosuppressive drugs. Now, how long do they have to be on immunosuppressive drugs? For a while or for life? For life. Is that a sure thing?
No.
Sometimes organ transplants fail in spite of everything. All right. So immunosuppression is the answer, but only sort of. Immunosuppression is a two-edged sword, is it not? You can't have a successful transplant of a homograft without immunosuppression. But if you're suppressing the rejection of a donated organ, aren't you, in a broader, general way, suppressing the entire immune system of the recipient?
Mhm.
Yes, you are. So this means what? That the recipient is more susceptible to failure of the immune system in other ways. Take, for an example, someone who has a successful kidney transplant. No problem with the transplant, but this person who never had a skin cancer now keeps having multiple skin cancers. Didn't happen before, but it only started once the transplant was done. So why? Likely answer, immunosuppression. They're more susceptible to infections and that sort of thing. So it's definitely a two-edged sword. So I think that's a good point here. So some terms. An autograft is a transfer of tissue from one body site to another in the same person. So someone who has a burn, significant burn, deep, third degree, that needs a skin graft, we can take skin from somewhere else on that person and graft it to that site once it's cleaned up and ready for a skin graft. That's an autograft. An isograft is the transfer of tissue from one member to an identical member, like identical twins. An allograft is another word for homograft. So this is between a transfer of tissue between genetically non-identical persons of the same species. So for example, your kidney transplant is that. Someone who has extensive burns who needs temporary coverage we can go to a skin bank and get cadaver skin as a temporary coverage. So it's transfer of skin from one human to another human. A xenograft, otherwise known as a heterograft, is a tissue transfer from one species to another species. So back to our burn example, if you've got an extensive burn and you don't have a lot of tissue for autograft, you need to buy some time to clean up the wounds, but keep a wound covered to keep down infection and to prevent protein loss and prepare the wound for an autograft. You can temporarily cover the debrided and cleaned up wound with pig skin, porcine skin. So this would be a xenograft. It's from one species, an animal of one type, to a human. Now, how long will that last? Well, it will be good for a couple of weeks maybe. And it's amazing how this can clean up a wound and get it ready for placement of the person's own skin. But it's not perfect, it won't last. It's just like a skin from a skin bank won't last either. It's just a temporary cover. All right, so on to antibody-mediated immunity. A little overview of how it works. Remember, we had our side-by-side demonstration. Here's the foreign antigen. Here's our general defense mechanisms. Here's our specific. But it has two arms, doesn't it? It has cell-mediated and antibody-mediated. So both arms can act against the same antigen, and commonly do. So here, we've got antigen presented to a lymphocyte that's specific for that antigen. With the, notice now, the assistance of helper T cells in the activation of that community of lymphocytes. They're becoming activated now. In the process, they're making some memory cells, and they're producing these cells. These are plasma cells. They come from these. Plasma cells are the ones that make the antibodies. The antibodies act against this antigen wherever it shows up in tissue fluids, body fluids. Whereas the T cells are acting against the antigen wherever it is in fixed tissues, and nucleated cells in fixed tissues. So you got them both working. Over here, the cytotoxic T cells killed by physical cell-to-cell contact. Over here, antibodies are acting against antigen by chemical reactions. Now, I think I told you at the beginning that cytotoxic T cells kill cells that contain foreign antigen. Antibodies do not kill, but they set up for destruction by other means. We'll look at that. Now here is another example of what I showed you just a while ago of how helper T cells are necessary for activation of lymphocytes. Here's a lymphocyte that has contacted the antigen it's specific for. But before it can become fully activated, it has to have the assistance of a helper T cell. This helper T cell bonds to it. This is this CD4 marker here. It's releasing some stimulatory proteins here. As a result of what this helper T cell does, this cell becomes activated, produces its memory cells, produces plasma cells, and antibodies that are specific against this antigen, wherever it shows up in body fluids. So, antibodies go after the obvious pathogen. They act in body fluids, blood, lymph, any other body fluids. The lymphocytes that are involved are B cells and helper T cells. So the B cells produce the plasma cells, which produce the antibodies. The helper T cells are necessary for co-stimulation, give them the okay to be activated. That's what they do. So again, antibodies do not kill or destroy foreign antigens, rather they disable them or prepare them for destruction by other means. So again, in review, here's the sequence. The B cell contacts a foreign antigen it's programmed for. It's activated. Well, I should say it's sensitized. This should be sensitized, really, I guess. And then the B cell ingests the antigen, presents antigenic fragments on its surface, class 2 type reaction. It must interact with a helper T cell. Now, the activated cells clone themselves and make more of their own kind that are specific against this one specific antigen. The B cells then differentiate into plasma cells. Some memory cells are made along the way. Plasma cells produce the antibodies. Memory cells held in reserve in case we meet up with this antigen later on down the line. In which case the response will be much more rapid and much more forceful. So, B cells respond to foreign antigen in body fluids, requires the assistance of helper T cell. They're activated, they proliferate, produce plasma cells and antibodies. Now, in this process of antibody-mediated immunity, you've got to remember that helper T cells are essential. Plasma cells are the ones that actually make the antibodies. Memory cells are held in reserve. Now, what is it that these memory cells actually remember about an antigen? Its specific molecular structure. That's going to be key, as I'll show you when we look at antibodies in a moment. So this is the thing in pictorial summary. B cell meets a foreign antigen. Sensitized, gets activated, clones itself, makes plasma cells make the antibodies. Now, this one here is showing you the primary response, which I've alluded to already. This is the initial encounter with the antigen. You notice that it did clone itself, made numerous plasma cells and antibodies. But because the memory cells are present, if we come in contact with this same antigen down the line, it could be a long time later, because we've already increased the numbers, the population of these memory cells that remember the specific molecular structure. If we meet that same antigen again, the response is faster and bigger, more powerful. All right, let's look at antibody structure. Antibodies roughly look like a Y. They have a stem region and two arms, like that. They're broken down into heavy chains and light chains. But this part out here is the variable segment. This part of the antibody out here is the part that recognizes the molecular structure of an antigen. And this, whatever antibody we're talking about, has a structure of its variable segment out here that is arranged in such a way. Remember the the three-dimensional shape of a protein determines its function. Remember that? All right, so the arrangement of the proteins in the antibody out here on these variable segments is specific for only one particular antigen. The only thing this is going to link up with is the antigen it's already specific for. Any other antigen, it won't touch, because there would be no binding of that foreign protein to the antibody. So all antibodies are proteins. They're in this class, immunoglobulins. As I showed you, they have the constant segments and the variable segments, but the variable segment is the one that determines antigenic specificity. And that's what's showing you down here. Another thing is that these antibodies do not bind to the whole antigen. There are certain antigenic determinant sites on the antigen. They bind to those sites. In other words, they don't cover them like a blanket. They latch on to the antigenic determinant site that's specific for that antibody. We have 5 different classes of antibodies. Now, the class of the antibody is determined by this segment right here, the stem of the Y. Not the prongs, but the stem. The stem determines the class. All right, so IgM, the Ig stands for immunoglobulin. So IgM are typically the first antibodies on the scene in response to an antigen. These happen to be potent agglutinating agents. They also happen to activate complement. Remember complement protein we talked about? IgA antibodies are found primarily in glandular secretions. They are helpful because they help prevent attachment of pathogens to mucous membranes and epidermis. Remember we said there's a class of antibodies that is in the mucous membranes of the tracts of our various systems. They're IgA class. The IgD is important in B cell activation. IgG are the most abundant. This is the biggest class right here. They are the main antibody of both primary and secondary responses. They also activate complement. They happen to be the only antibody that crosses the placental barrier. And then we have IgE, found in skin, mucous membranes of various tracts. Allergic reactions typically involve these antibodies right here, IgE class. These are some of the geometric designs of our various classes. This IgM, for example, has multiple antigenic determinant sites, or multiple variable segments. This kind of antibody is a very good agglutinating agent because, like in a transfusion reaction, when you have antibodies attacking surface antigens of mismatched red cells, these reach out and catch on to an antigen on a red cell here. And over here you might have another antibody just like this that catches onto it too, another site. And if you repeat this in multiple times, that's what causes the clumping, because you've got pieces of antibody catching on to various red cells, and they agglutinate or clump these cells. And these are the other types right here. All right, now we said that antibodies do not kill, but they immobilize and set up for destruction by other means, right? So here are the ways they do that. neutralization. What this means is that the antibodies block specific sites on viruses or bacterial exotoxins, and these bound pathogens are eventually destroyed by phagocytes. Agglutination, what I just described, what you see in mismatched red blood cells, for example. This is cell-to-cell clumping. Precipitation is a process like this, except it's chemicals. It results in precipitation in solution. And then we have complement fixation and activation. The stem regions of antibodies are responsible for this. And this process also stimulates inflammation and phagocytosis. So let's look at a pictorial display of how this works. So here's antigen, here's the antibody attacking it. We have an antigen-antibody complex. So these antibodies will inactivate by neutralization, agglutination, precipitation, or activation of complement protein. Now, the antibodies haven't killed any cells yet, have they? All right. But once this happens here, any of these three ways, this leads to inflammatory reaction and phagocytosis. So phagocytes come along and gobble those up. Over here, if complement is involved, remember complement protein leads ultimately to puncturing of holes in cell membranes of affected cells. So in this case, we have class 1 type, classic type of complement protein action resulting in cell lysis. So it's not really the antibodies per se that lead to the killing of the cells, it's the complement protein that they brought on that led to the killing of those cells. All right, now I mentioned before that excuse me, when we come in contact with a new foreign antigen that the body has not seen yet, the initial response to that has a lag time. In other words, in order to build up an adequate titer of antibody, it's going to take a week or two. So there's a lag time. So the primary response is slow. We have to build up a sufficient titer. Takes 1 to 2 weeks. The lag is due to the time it takes to proliferate these B cells and make the plasma cells and then make the antibodies. The IgM class is the first on the scene in the primary response. The IgG class is slower, but they are the larger representation once the process gets going. If for some reason the exposure to the antigen stops in this process, the whole thing falls off. We don't get the full effect. Now here's the secondary response. Remember that when the primary response occurred, we were also developing memory cells, weren't we? That were held in reserve. If down the line, could be weeks, months, years later, we meet this same antigen, we already have a population of lymphocytes that's been cloned that recognizes this antigen, and they're going to pounce on it right away. So the response is going to be rapid, and the extent of the reaction is going to be more severe. So it's fast. Quick buildup of antibody titer. Those blood levels are going to remain high for a longer period of time. This is the way it looks if you graph it out. So let's say we meet our new antigen, foreign antigen, right here. This is days here this is antibody titer here. So it's going to take a week or two to get a maximum titer to handle that initial exposure. And then the titer is going to fall off as we handle the situation. Down the line, now this is just a few days down the line, but it could be months or years down the line, we meet the same antigen. Now look at the difference. The antibody titer is much higher and it happens much quicker. Now in the meantime down here, we come in contact with a different foreign antigen. We've got to go through the same process that we meet for the first time. We've got to go through the same thing here because of a different antigen. So specific antibody communities or neighborhoods of lymphocytes react against specific antigens. Now, sometimes things go wrong in the immune response. So sometimes our immune cells can attack our own cells. Not good. Not good. But it can happen. So an autoimmune disorder is a malfunction of the immune system that's characterized by a loss of the ability to distinguish self-antigens from non-self antigens. It causes the body to produce antibodies against self-antigens that are perceived by cells of the immune system to be foreign to it. Now there are different possible explanations for this autoimmunity. One is that some self-reactive T cells and B cells have escaped elimination in the thymus. Well, actually I shouldn't say B cells because it's T cells in the thymus, but some of those T cells, for example, they slip by in that thymus testing out. And some of the B cells in the bone marrow slipped out of the immunocompetency testing, and they're now attacking normal cells. Sometimes we might have new self-antigens that were not previously exposed to the immune system. Sometimes we might have mistaken identity. In other words, certain tissues of the body have a molecular structure that reminds certain cells of the immune system of foreign antigen. And so they attack normal cells. This, for example, is what is thought to happen in multiple sclerosis. In multiple sclerosis, immune cells are attacking myelin sheaths of nerves and causing short-circuiting of nerve impulses. So this is sporadic, it's intermittent, but it's attacking myelin sheaths of nerves. It's thought to be a case of mistaken identity.
Is that in arthritis too?
I'm not sure whether it is or not. It might be. It might be. Immunodeficiency diseases are caused by any condition that mutes or disables the immune response. A typical example of an immunodeficiency disease is HIV/AIDS. Because the virus is attacking the immune system directly, and specifically, in this case of HIV, it's attacking the helper T cells. Remember what we said about helper T cells? They are required for both the function of T cells and B cells. Hypersensitivities, I'm not going to go into this a lot, but hypersensitivities are what result in allergies. Allergies are a type of immune reaction. They don't confer immunity, but they cause a lot of aggravation and unpleasantness, don't they? All right, so the immune system is causing damage because of a perceived threat, but otherwise harmless. There are several types of hypersensitivity: immediate, subacute, and delayed. They're distinguished by their time course and whether B cells or T cells are involved. We won't go into all of them, but let's just talk a little bit about hypersensitivity, the type 1 hypersensitivity. The thing about this is that in the initial meeting, we are sensitized to an allergen, but only sensitized to it. There's no reaction yet. So an allergen is introduced. Antigen-presenting cells present this to helper T cells. Those helper T cells are sensitized. Now, the B cells then produce IgE secreting plasma cells, which produces IgE antibodies on the stem regions. They attach to this kind of cell, basophils and mast cells. That's where it stops with the initial meeting. We sensitized when we got no reaction. The host is sensitized to this particular allergen, this particular foreign antigen. Now, later on, we meet up with the same allergen again. This thing is already set up to act because you've got mast cells and basophils attached to the stem regions of antibodies. And now they're released into the bloodstream, and these kinds of cells here secrete histamine, a vasodilator, because an inflammatory reaction. So an allergic reaction is just one type of inflammatory reaction. So this is the way it works, right here. This is the sensitization stage. We're only setting it up. Now we meet the same allergen again, and we get the local reaction. We get swelling, mucus production, itching, coughing, runny nose, whatever. Depends on where the allergen is attacking. If it's attacking the intestinal tract, you get intestinal disorder, nausea, vomiting, diarrhea, and so forth. But again, most of these hypersensitivity / inflammatory reactions are localized reactions, aren't they? Localized. Sometimes, however, this is a systemic reaction. So now, the reaction is not localized, it's systemic, and you've got massive peripheral vasodilation. What happens to blood pressure and blood flow when you get massive peripheral vasodilation quickly like that?
Drops.
Drops, doesn't it? So, that kind of reaction is known as an anaphylactic reaction. It's potentially fatal. Some people have these, like some people who are, say, sensitive to bee stings. Some of these people will carry around with them some medication, like epinephrine. Epinephrine is an adrenergic hormone, isn't it? Mm-hmm. Epinephrine does what to the heart?
Speeds it up.
Speeds it up. It does what to systemic blood vessels? Constricts them. What's the net effect of that? Vasopressor. Yeah. Now, if you're just having a local allergy attack, say an upper respiratory thing, what do you often take for that?
Antihistamine.
An antihistamine. What caused the problem to begin with?
Histamine.
So you take an antihistamine to try to stop all that vasodilation. Another thing that, sometimes is required in an anaphylactic reaction is not just epinephrine, but it may take steroid therapy. You know, the administration of steroids. We're going to find out later that steroids, one of their effects is suppression of the inflammatory response. All right, the others you can read about. This just summarizes the type 1 hypersensitivity, local reaction versus systemic reaction, right here. These other types are related to whether B's or T's are involved. I won't go into that. You can read about that. Now, in the little bit of time we got left, let's talk about this HIV/AIDS briefly. Just summarize this. I told you that this will be on the lecture exam. Part of the exam is going to be some of these exact questions, right? So, you can find the answers to these questions in this little handout I gave you on HIV/AIDS. So once you nail that down, you can answer these questions. So, HIV is a virus. HIV stands for human immunodeficiency virus. You got that? Human immunodeficiency virus. AIDS is an acronym that stands for Acquired Immunodeficiency Syndrome. Now, the HIV virus is what is known as a retrovirus. Viruses do not have their own DNA, and they commandeer the DNA of the host. And that's what HIV is doing here. So it's using host cells to make more of itself. That's what a retrovirus is doing. Now, it asks the question, HIV exerts its primary effect on which of the following? Which one?
Helper T cells.
Helper T cells. Exactly. The specific class of T lymphocytes that is unaffected or relatively unaffected by HIV is the regulator T cells. AIDS is a leading cause of death in what age group in the U.S.? 25 to 44. In the U.S., the most common mode of transmission of HIV is sexual transmission. The second most common cause is shared IV needles, not IV needles themselves. Shared IV needles. All right. After the initial infection by HIV, when does the infected person typically develop the first clinical illness?
After 5 years.
In a few weeks. Look at that diagram in the back here. This diagram back here shows you the clinical course of a person infected with HIV. So, in this column here, we've got the CD4 T cell count. Here, we've got time. Okay, so this dotted line represents the CD4 T cell count, and it's going to correlate with the appearance of symptoms and signs in the infected person. But here is the initial event right here. Right here. The person doesn't get sick right away. But in a few weeks, they will develop an illness that looks a lot like flu. And its associated symptoms. And then they get over that. And following that is gonna be a latent period that will go on for some years here. So we got kind of a flat line down here. In the meantime, notice that the CD4 T cell count is progressively dropping as we go out in time. Notice that these lines cross as the CD4 T cell count drops. Now, the constitutional symptoms start showing up. They're getting sicker and sicker because they're losing their helper T cells out here. At first, they develop constitutional symptoms of various types. You can read about those. As the CD4 T cell count drops. Now, CD4 is denoting what? Helper T cells. The marker on helper T cells, CD4. As the CD4 T cell count continues to drop progressively, not only do we have these constitutional symptoms appearing first, but now these people are getting sick with things that a normal person could easily fight off and that never bother a person with a normal immune system. Those kinds of things that are happening now, right up here, are called opportunistic infections. These are infections with things that the body normally and easily fights off. You never give them a thought in a normal person. Now they're sick with them.
Like thrush or something?
Huh?
Like thrush?
Could be that, but that's going to usually occur back down here with fungal infections. But now you've got, say, a lung infection up here called pneumocystis carinii. That's an infection in the lungs. Normal people, that's not a problem. But now they're susceptible even to things like that. They start developing neurological symptoms. Okay, so they also can develop dementia in here. Okay, so you're approaching death when you start developing opportunistic infections. So that's what this graph is showing you right there. So that answers some of the questions along here. How do you test for HIV? The initial test is an acronym, ELISA. Okay. And the follow-up test, if the ELISA is positive,